Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. In the previous lecture, we have discussed in detail about the life and how the life was originated onto the earth. So in that context, we have discussed about the many theories which were explaining the origin of life onto the earth. We have discussed about the theory of special creations. We discussed about the theory of spontaneous generations, which actually believes that the non-living matters are giving rise to the different types of organisms. But that theory was incomplete because it has not been based on the experimental evidences. And there were critical experiments which are being performed by the Reddy Spanagili or the Louis Pasteurs to disprove that this is actually happening in the current environment. And then later on, there were many other theories like the theory of catastrophisms, theory of cosmozoics, theory of eternity of life, and ultimately, we have also discussed about the theory or modern theory or the chemical theory of the origin of life. And chemical theory was the first theory which was based on the experimental evidences where the Stanley Miller and Urey has done the very meticulously and very systematic experiment to prove that if you take the inorganic substances and if you if you run them under the primitive earth conditions which was reducing in nature, then you could be able to develop the biomolecules. And that was the first evidence that the life could be originated from in the primitive environment because of the chemical interactions or the from the inorganic molecules. And then they have actually proposed the event of the experiments, how the different event could have happened onto the primitive earth, and that's how they have said that ultimately the inorganic substances are going to react with each other to give you the simple molecules, which they have actually demonstrated by their classical experiments. And then they have said that these simple molecules are going to react with each other to give you the complex molecules. These complex molecules will then aggregate and give you the coisorbates, and the coisorbates are actually, they will develop the tendency of a living organisms. If you recall, we have discussed that what is the living organism. Living organism is an organism which could be able to run its metabolism, which could be able to produce the energy, and which could be able to repair. And that was all the properties were present in the coisorbates. But ultimately, the coisorbates have started, acquiring the biomolecules from the primordial oceans, and then it has actually acquired the nucleic acids and proteins and lipids, and ultimately the coisorbates were developed into the first cell. There's a complete breakage, or there is a gap what will explain that how the coisorbates are developed into the first cell, which could be able to run its metabolism, which could be able to draw the nutritions and so on. And one of the question which they were be able to solve, that why these things were not happening in the current environment, and why the theory of spontaneous generation could not be able to be successful, because the current environment is deoxidizing in nature, and because it is oxidizing in nature, as soon as these inorganic substances are trying to react spontaneously, trying to react with each other to give you the simple molecule, they are getting oxidized, and because of that they are not being able to form the complex molecules. So these are the things what we have discussed, and we have discussed ultimately that the simple cell is being formed. But as we know that that simple cell gradually being developed into the multicellular organisms, and then the multicellular organisms develop into the higher class. So the question in today's class what we are going to discuss is how and what is the mechanism of these simple primitive cells being evolved into the multicellular organisms or the higher organisms. What we have discussed? We have discussed that the life is originated as the primitive cell with an ability to replicate, absorb the nutrition, and it can be able to repair the damages, right? Although we are having a very big gaps how the coisorbates are being developed into the primitive cell, but that is we still probably have no conclusive answer to that. These single cells are the starting material to form the multicellular system, and eventually to develop the organisms with the tissue and organ systems. So the changes, the progressive advancement of these organisms is by a process to acquire the traits so that it can be able to adapt to the new environment is known as the evolution. And Aristotle, which was considered to be the father of biology, considered the evolution as the ladder of chain. So where he, or the scala naturae, involving the hierarchical linking of the series of the forms. So what you have seen here is actually the ladder of chain, where the organic matters are being placed at the bottom, right? So what you see here is the inorganic matters which are being placed at the bottom. Then the inorganic matters are being developed into the lower plants. That lower plants are going to be developed into the higher plants, and then these higher plants are being developed into the lower invertebrates like jellyfish and sponges and all that. And then these jellyfishes are being developed into the insecta and archaeodes. Then these insects are being developed into the snails. Then snails are being developed into the crab, and then crabs are being developed into the squids or octopus, and then octopus are being developed into the fishes. Then fishes are being developed into even the more advanced fishes, and then the fishes are being developed into the reptiles, and the reptiles are developed into the birds, and then it will be developed into the mammals, and ultimately he has kept the humans on the top, right? This is simply by not being based on the experiment. These are all simply based on the observation as well as his own personal opinion that this could be the ladder of chain in which the organism are being evolved. But the question comes, what is the evidence that the evolution is really happening? Because until unless we cannot have the experimental evidences, we cannot have to believe this particular type of scheme, right? We cannot say that lower plants are being developed first and the animals or the plant or humans are being developed at the later on, right? So then people came up with the idea of chemical evolution. The term evolution referred to the changes from the one form to another form, and that form is always for the advancement. So the change in living organism with the time to known as the organic or the biological evolutions. The process of evolution can be understood from the fact that the unicellular organisms appear first, right? Unicellular organisms appear first. The simple multicellular organisms came later, and later they were developed into the complex multicellular organisms such as the seed plants as well as the vertebrate animals. The fishes were the initial early vertebrates, and it gradually changed to form the amphibians and so on. These amphibians has produced reptiles, and that evolved further to give the birth and the mammal. This hierarchical linking of different species is considered as the ladder of chain by the Aristotle. In the same series, the mammal have evolved to the human, evolving the ape-like primate by acquiring the changes over the course of time. So the chemical evolution or the normal evolution is actually the gradual change in the animal forms or the organism form so that it can be able to acquire new and new traits, right? You might have seen the evolution is either be very slow, right, where the monkey is being evolved into the humans, or it could be very very fast, right? You might have seen the adaptations in bacteria and how you develop a single drug, right? Suppose you take a bacteria, right? For example, the mycobacterium tuberculosis, right? And if you treat it with the drug, the mycobacterium tuberculosis actually died, right? But in a due course, the mycobacterium tuberculosis actually do the changes, do the changes in its cellular body, right? And that's how it actually comes up with the multi-drug resistance MTB or drug resistance MTB, right? So because of that, it actually forms a drug resistance TB, and this is called as the evolution, because now this is not going to be killed by this particular drug. And this is exactly what is happening. Like once you are actually putting a challenge, or once you are putting a challenge to a particular organisms, it will actually try to come up with the ways in which it will actually going to overcome that particular problem. And that is the main basis of the evolution, right? That it has to overcome, it has to acquire the additional traits. For example, in the primordial oceans, the primitive cells is formed, which is the unicellular cell, right? And that unicellular cell were having the deficiency because the unicellular cells cannot actually get the support from the neighboring cells because it is actually a unicellular cell. So what he thought is that let's have a multicellular cell, right? If you go to the multicellular system, so in the multicellular system, the advantage is that first of all, you can actually do division of labor. You can say, oh, you will do this, I will do this, and all that. And on the other hand, even if the one cell is going to be under the threat or it is actually going to be die, the organism will still be able to survive because the organism is actually going to overcome that particular type of problems. Similarly, once the multicellular system, it actually could develop into the organ system, and we know that the as you go along the this system, you are actually acquiring more and more complexity, and more complexity always comes up with the additional features, and that's why the people have observed that the organism first appeared as a single unicellular organisms like the protozoa, right, amoeba. Then it goes into the multicellular stage where it has actually developed the sponges and coelentrates and all those kind of hydra, like for example. All these information came because we have classified the organisms and we understood why it is happening so, right? And then these multicellular organisms develop into a system where they can actually have the different types of organs, for example the platyhelminthes, and then the platyhelminthes eventually developed into more advanced system where you don't have one single organ but you have the organ system, like for example the humans. So humans have the different types of organ system, like you have the liver, we have the kidney, we have the pancreas, we have the stomach, we have the different types of organ systems. So every organ system is doing its job, but it also trying to talk to its neighbor, right, trying to talk its other organ system. For example, if you talk about the digestion is actually not happening because of the alimentary canal, right? Digestion is also happening because it is getting the support from the other organs like pancreas, it is getting the support from the liver, it is getting the support from the some more other system, right? So organ system is the most advanced system what is present in the organisms, right? So, and that helps that particular organism to face the different types of the challenges. So that is what it been considered by the Aristotle, and that's how he has prepared a ladder of chain. So what you see here is a ladder of chain which actually been designed based on the who can do what, right? It's not been the same way in which the organism are being evolved. This is based on the Aristotle's understanding that okay, this particular organism is having these kind of deficiency, whereas these deficiencies are not present in the other organisms. So he has kept that particular organism into the lower level and he has kept the other organism. For example, he has kept the human on the top, right, because it believes that the humans have all the tools and all sort of systems so that he can actually be able to overcome all sort of problems, right? So that's why he has kept the humans onto the top, right, whereas he has kept the lower plants and all those kind of thing at the bottom, right, because they cannot do many of the things which higher plants or the organism could be able to do. So this is all about the chemical evolution and that okay, there is a evolution happening. But once you talk about the science and when you say that evolution is happening, you always looking for the experimental evidences and you are looking for the some sort of evidences to prove that there is actually a system where is a through which the lower invertebrates develop into the higher vertebrates, and then higher vertebrates develop into the different types of animals like the mammals and birds and snakes and all that, right? So what is the evidence, what is the chemical evidence that the evolution is really happening? So now the question is that what are the scientific evidences that the organisms are evolved from the previously existing organism, which means whether there is a evolution or not, right? That we have to answer. So if you study the physiology, anatomy, development of different organisms, give the clues about the several similarities between the related organisms with the selected differences, right? So The correlation of the differences within the related organism has allowed to identify the properties used to study the evolutionary stages of an organism. These evolutionary evidences are as follows, right? So if you study the physiology, if you study the anatomy, if you study the how the different organisms have the different types of developmental stages and how they are actually being similar to the other organism, but they still have their own peculiar features, that all if you correlate, it will actually going to give you a detailed the relationship between the different organism, and that's how you can be able to study or you can be able to understand the evolutionary stages of an organisms. These evolutionary evidences could be classified into the 3 points, right? It can be either related to the morphology or the structural evidences. It could be because of the embryological evidences, or it could be because of the paleontological evidences, which means where you are actually going to study the fossils, right? So fossils are also a very good evidences that actually is going to prove that who came what time, right? Within the morphological and structural evidences, you can have the anatomy, right? You can have the evidences which are actually going to say how the body is being organized. Then you can have the similarity within the physiology, so you can have the homologous organs or you can have the analogous organs, and then you can also be able to study how the physiology is being modified by the gradual modifications. So and then you can also be able to study the connecting links. So connecting links are the organisms which are actually being connected between the two different organisms. So they have the features which are common to the two different features. So let's discuss each of these evidences and try to understand that how the scientists have come up with the idea that the evolution is really happening, and there is a series or there is a event, there is a scheme through which these organisms are evolving. One scheme is anyway been proposed by the Aristotle, where he has proposed all the linking of all these organisms and ladder of species he has proposed, but that was not on the basis of the experimental evidences. So let's first discuss about the morphological and structural evidences. So as far as the morphological and structural evidences is concerned, if you do the comparative study of the morphology, which means and the anatomy of organism, indicate that the few of the features are similar. These are as follows, right? So if you try to see the morphological and structural evidences, what you can see is you can be able to identify the similarities between these features, or you can be able to understand the differences, right? So if you go with the similarities and the differences, what you can be able to understand how these organisms could be evolved. The first is you can go with the body organizations. The body organization of different organism is evolving over time with the different level of organizations. The unicellular organizations with the single cells are the most primitive body organization, followed by the cells to arrange to give rise to the tissue. The tissue gather to form the organs, and the organ cooperate to form the organ system. For example, right? So this is what we are talking about. Initially, we were having the unicellular organisms. These unicellular organisms then, differentiated, and they realized that okay, we are actually being at the danger, because as soon as we are actually getting any kind of scarcity, for example, if there will be a food shortage, if there will be a food shortage for a unicellular organism, he will die because there is no alternate source, because he cannot. But once it develops into multicellular organisms, or it can be developed into a tissue level organisms, so what will happen? So here you have the single cell, so you have one cell, right? Suppose you have developed into a tissue level organizations and you have the 100 cells, right? And again, there will be a food shortage. So what will happen? These 100 cells, out of these 100 cells, what will happen if suppose the 10 cells are going to die, okay? These 10 cells are actually going to die, and they will be having the organic matter, right? This organic matter, that organic matter is actually going to be utilized by the remaining 90 cells. Number one. Number two, since it goes into a stage where it has the 100 cells, and if there is a threat coming, like for example, if there is a prey which is actually trying to kill them, right? You have the 100 people which are fighting. So you can imagine that if you have single person fighting with the enemy, it is difficult, it is not possible to win. But if you have the 100 people fighting with the same enemy, there could be a possibility that you may be able to win, because you can be able to fight with the full strength, right? So you can be able to get protected, number one. Number three, that is the protection, right? Even if the running the physiology also, if you have the 100 cells, these 100 cells could actually be able to do things more efficiently, because you can be able to do an exchange of material. For example, if you have a single cell and you cannot be able to even take care of the byproducts of the metabolism, the byproduct is actually going to ultimately kill the unicellular cells, right? Whereas if you have the 100 cells, the byproducts could be scavenged, byproduct could be scavenged by the other cells. And because when you have the 100 cells, some cells are actually going to be the new, some cells are going to be old. So these new cells are going to be more efficient in terms of performing the functions, whereas the old cells are actually going to less efficient. So these old cells will still survive because they are having a support of the new cells. Similarly, when the tissue is going to develop into the organ, it is actually going to come up with the more and more sophistications, because the organ is going to be more organized. So it is going to have the different types of tissues, and that's why there will be a further level of division of labor, right? And once the organ is going to develop into the organ system, then you are actually going to have the support from the different organs. So that is actually going to make the things more and more systematic and sophisticated. For example, in the humans, we have the different types of, for example, in a single cell, the single cell is performing all the functions, whether it is respiration, whether it is food intake, whether it is taking care of byproducts, whether it is the water imbalance, and so on. Whereas in the case of humans who have developed the organ system, you have the heart, right? You have the heart, or the circulatory system, which is actually going to take care of the circulation of the material or distribution. Then you have the lungs, which is going to do the respiration. Similarly, you can have the liver, you can have the kidneys, and all those kind of things. So all these organ systems have their own dedicated functions, but that does not mean that these organs are not going to talk to each other. Heart is always going to listen to the brain, and liver is also going to do coordination. So there will be a coordination between the different organs, and that's why it is going to have the organ system. So that's why the organ system is the highest level of the organization which is present. Now let's see the example. So amoeba is unicellular, right? Sponge is multicellular, or the tissue level organizations, but these cells are not organized into the tissue to exhibit the cellular level of organizations. Whereas in the case of coelenterate, like the hydra, cells are organized to form the tissue, but later do not form the organ. This is a tissue level organizations. Similarly, in the platyhelminthes and higher animals, the different types of tissue give rise to the organ system of organizations. So if you go with these kind of evidences, where you have the body organization as a criteria, you understand that probably the living or unicellular organisms appeared first, then they will be developed into the tissue level organization, then they develop into the organ, and then they will develop into the organ system. So if I go with only this particular evidence, I will say that the amoeba is being evolved into the sponges, and then the sponges are being evolved into the coelentrates, and then coelentrates are actually coelentrate, and the coelentrates are going to develop into the platyhelminthes, and the platyhelminthes are actually going to develop into the higher vertebrates, and then subsequently into the vertebrates. Same is actually also be true for the plant system. Like the plants, you have the unicellular plants like the algae, then algae is actually going to develop into the multicellular plants like the fungi, then the fungi is going to develop into the bryophyta, And then the bryophyta is actually going to develop into the pteridophyta, and the pteridophyta is going to develop into the gymnosperm. These are the lower plants, and then the gymnosperm is going to develop into the angiosperm. So if you go by the body organizations, you could get the clues and as well as the scheme through which the organisms are being evolved from the pre-existing organisms. Then we have the homologous organs. That is another evidence. The organs of the different species of a common descent which looks different and perform different functions but has the similar structures, similar topographic origin and similar embryonic origin are called as the homologous organs. Homologous organ means the organism's anatomy is going to be identical, but they are actually going to do the different functions. Okay, so they will perform the different functions, but their anatomy is going to be different, same, right? We have couple of examples. The homology is based on the divergent evolution, so it will be actually be based on the divergent evolution, right? Because of the adaptations, so it's this single organisms got evolved into these three organs, but he has not changed the anatomy, he has only changed the way these organs are going to be utilized. One of the classical example is the forelimb in the vertebrate animals. So we have the forelimbs in the man, cheetah, whale and the bear, and they are of different shape and perform different functions. For example, these are used for grasping objects. So these are the four examples. You have the hand in the case of man, then you have the cheetah, right? So you have the forelimbs in the case of cheetah, then you have the whale, and then you have the bat. And you know all these four vertebrate animals are different, right? For humans, they are actually using the hand for holding the objects, right? So grasping the object, whereas the same forelimb is going to be used for running in the case of cheetah, whereas in the case of whale, that particular forelimbs is going to be converted into the limbs, right? So swimming in the whale, and the same is also being used for the flying in the bat. In each case, the structure of the forearm has similar plans, like upper arm having the humerus, followed by the radius and ulna, and the hand with the carpals in the wrist. So if you see the anatomy, right, its anatomy remains the same, right? You have the humerus, you have the radioulna, so you have the radioulna, and then you have the hands, right? So here also you have the humerus, you have the radioulna, and then you have the hands, right? But the functions are different. Functions are that in the case of human, it is for holding the object, whereas in the case of cheetah, it is for running. So it is being adopted accordingly, right? It is being adopted to give that particular type of functions, whereas in the case of whale, it is for the swimming, and this bat, this actually got the membrane. So last part is actually getting the membrane, and because of that it got converted into a wing, right? And that's why it is being used for the flying. All vertebrates have the basic similarity in the structure of their forelimb due to their origin from a common ancestor with the 5 digits. So that's why all these animals are actually being originated from the single ancestor, and that ancestor is actually having a pentadactyly feature. So pentadactyly means the organism which will actually going to have the 5 figures. Okay. Then we have another example. So another example is you have seen the thorn and the tendrils. Thorn is actually being used for the protection in the plant, right? Because it protects the plant from the from the herbivores, like for example the cow or buffalo, all these grass eating plants, right? So they will be having the thorns, so that when they try to eat, these thorns are actually causing the injury, and that's how they will be get protected. Whereas the tendrils are actually being used for the climbing, right? So they will be found in the creepers, and they will be used for the climbing. So the thorn in bougainvillea and the tendril in the passion flowers are the homologous organ in the plant, because they are actually having the similar kind of anatomy, except that here you have the pointed knobs, whereas in the case of this it got converted into a spiral spring like structures. They look different and they help in the plant in the climbing, but both arise from the auxiliary position and are modified branches. So these are actually being modified branches. One is used for the climbing, the other one is used for the protection from the herbivorous or protection from the cattle. Then we have the analogous organs, the organs which perform same function and look similar but are quite different in their structure. So these analogous is actually exactly opposite. So they actually do the same function. So they have the same function but they are different in terms of structure. Which means, if you remember, in the case of the homologous organ, it was actually telling us the divergent evolutions, whereas the analogous organs are going to tell us the convergent evolution, which means these two are actually being converged evolutions. So if you see an analogous organ, it is actually going to tell you the convergent evolution, because the structure is more important than the function, because the function is very can be based on the adaptation into that particular environment. So the example is, there are several examples, like for example the insects and the winged bird. So you have seen the wings in the butterfly, right? So this is the butterfly, and you might have seen the wing in the case of bird, right? So both are actually the analogous organ, because they both are being used for one function, that is the flying, right? They both are being used for the same function like the flying, but their anatomy is different. You see the anatomy of the bird wings, and you see the anatomy of the flies, right? They are very different, right? So the wings of the bird and the insects are the analogous organs. In both organisms, these organs are used to fly in air, but they are different in terms of their structures. Insect wing is an extension of the integuments, whereas the bird wing is formed of the limb bones converted with the flesh, skin and the feathers. So they are different in terms of their origin. This is coming from the integuments, whereas this is actually an extension of the forelimb, right? So where you have the feathers and all those kind of thing, and that's how it actually get the ability to fly, right? So it actually be able to change the air, and that's how it actually be able to uplift the birds. Then we have the another example where you have the fin and the flippers. The pectoral fin of the fishes and the flippers of dolphins are flattened organ used for the swimming, but both, so here in this case we have the one function, which is the swimming, right? So for swimming, these two fishes are actually using their fins, and they have the fins and the flippers. So they are actually doing the same functions, but their anatomy is different. So but their structure is different. The flippers are the modified pentadactyly forelimb, whereas fins are the pentadactyly. So these two are actually being formed from the different origin and different structures, but their function is same. So the other evidence for the within the category of the morphological and structural evidence is the gradual modifications. In several cases, the organs or the tissue exhibits the gradual modification during the course of the organic evolutions. For example, the heart. Heart is a classical example, because heart was initially being two-chambered, right? Then that got converted into three-chambered, and then that got converted into four-chambered. Within the two-chambered, you have the auricle and ventricle, right? Within the three-chambered, you also have the auricle and ventricle, and so on, right? So you have, see here, if you have the two-chambered, it will be one auricle and one ventricle, which is actually be present in the fishes. If you have three chambered, it is actually two auricles and one ventricles, which is actually been present in the amphibians. And then there is a pseudo four chambered heart, which is having the two auricle and partly divided ventricles, which is in the reptiles or the snakes. And then you have the completely well developed four chambered heart in the case of the higher reptiles like crocodile, birds, and as well as the mammals. So between this you also have the four, which is partially coated, right? So this is what you said, right? This is actually going to be a intermediate stage, right? And this is the two chamber, right? So this is having only two auricle and as well as the ventricle, right? And before this, there could be one chambered heart which was formed, right, which was the primitive heart probably be formed, and that got split into two chambered. And that's so if you see like cockroach or lower invertebrates, they don't have the heart but they have a pumping system, and that is not having a chamber. So this is not, so there's no one chambered heart is present in the any animal, but the system what is present in the lower invertebrates are like they have a pumping system, but that is not having a chart, right? So you can imagine that two chambered heart, right? Two chambered heart has auricle and ventricle. Now what is the disadvantage? Disadvantage is that it is actually not going to avoid the mixing of the blood, right? You know that the ventricle is actually having the pure blood, whereas auricle is having the pure blood, whereas the ventricle is having the impure blood, right? So if you don't have the chambers, you are actually going to mix these blood later on, because the auricle is actually going to receive the the pure blood from the lungs, but and then it is actually going to pump that blood into the body. So once you go from the two chambered to three chambered or three chambered to four chambered and so on, that is actually going to increase the efficiency of the working, right? You can be able to very precisely control which chamber is going to accept the deoxygenated blood, which chamber is going to give you the oxygenated blood, and so on, right? And that's how you can avoid the mixing of the blood, and that's how you can be able to perform the functions properly. So if you see the gradual modifications, this is just one example where we have taken an example of heart. It actually says that initially the fishes are being developed, right? So fishes, then that fishes are actually given rise to amphibians, then amphibians are being developed into the lower snakes, right, or the lower reptiles. Then the lower reptiles are going developed into the higher reptiles, like higher reptiles where you have the crocodiles and all those kind of thing. And then the higher reptiles got into the mammals or birds. So if you see, all these evidences clearly says that the ladder of species what is being proposed by the Aristotle was not completely correct, right? It was based on some assumption, but here you see that if you see the ladder of species, this is not a sequence in which it has been placed, right? It is being placed in a different sequence where the birds are placed lower to the reptiles. Then we have the another advantage, the other example, other advantage is the connecting links. So what is the connecting link? For example, you have three organisms, right? So you have organism 1, you have the organism 2, and you have the connecting links. Now this connecting link is actually going to diverge into these two species. So it is actually going to have the mixture of properties what are being present. So if this is, suppose this is A, this is B, it is actually going to be AB. So this, first of all, this organism is being evolved, right? And then depending on the conditions, depending upon the environment, depending upon the other kinds of adaptations, this particular thing has given up the B. So if there will be a given up of the B character, it will develop into organism A. If there will be a giving up of the A, it is actually going to develop into the organism B, and that's how the connecting link is actually going to tell you that okay, these A organisms and the B organisms are related to each other. They are actually going to have their common ancestors. You might have seen that kind of common ancestor is also exist even within the different different races within the humans also, right? The living organism exhibit character of the two different groups of organism and known as the connecting links. There are few selected example of connecting links, and few examples are as follows. Your example is a euglena. Euglena has the dual character of the plant and animal. So euglena is actually going to be a connecting link between the plants and the animal, and it is considered that probably the euglena like molecules are being evolved, which was actually having the chloroplast at the one side, and they also have the system so that they can be able to catch the prey. It can perform the photosynthesis through the specialized chloroplast, and it can perform the contractile vacuole mouth and binary fission, and it can actually be able to eat just like the animal, and that's why this is considered to be a connecting link between the plants and animals. Then we have an example 2, which is the peripatus. So it is an example of connecting link between the arthropoda and the annelids. It has worm-like body, so that is a character of unjoined legs, which is similar to the arthropoda annelids, whereas it has claw, jaws, trachea, and the dorsal tuberal heart as an arthropoda. So this is what I was talking about, dorsal tuberal heart, which was present in the cockroach and which is actually pumping the blood, but that is a single chambered organ, the tissue, and it does not have the well defined muscles and other things to pump, but it is actually distributing, and it is considered to be heart like structure. So this is what, right? So peripatus is an example of connecting link between the arthropoda and annelida. Similarly, you have the egg-laying mammals. You know that the mammals are actually giving rise to the babies, right? But in this case, we have the egg-laying mammals. So egg-laying mammals are the connecting link between the reptiles and the mammals. For example, you have the duck-billed platypus. So this is a duck-billed platypus. They have a few mammalian characters such as hair, mammary glands, we have the diaphragms, whereas it lay eggs with yolk and egg, which is similar to the reptiles. So if you recall, when we were talking about the classification of the living organism and we were talking about the features of the mammalian system, we said that there are 7 characters which should be present onto the mammal. Like one of the character was the hair, the mammary glands, and the well developed the respiratory system. So these are the 3 characters which were present in the duck-billed platypus, but they were also going to lay the eggs with yolk and eggshells, which is similar to the reptiles. Then we have the several more examples also of the connecting links. For example, we have the neoplina, which is a connecting link between the annelids and the mollusk. Then we have the balanoglossus, which is a connecting link between the non-chordate and the chordate. So I'm not going to discuss about the multiple features. The only idea was to just to give you that the connecting links are actually a very crucial evolutionary evidence that the two organisms actually been evolved from that particular organisms. Then you have a chimera. It is a link between the cartilaginous and the bony fishes. Then we have coelacanth, which is actually a link between the bony fishes and the amphibians. And then you have spendon, which is a connecting link between the amphibians and the reptiles. So connecting links clearly highlights the fact that the different organisms are evolved together from a common ancestor, and that is a very strong evidence that the evolution is happening from the earlier primitive form of these organisms. Then we have the embryological evidences. So embryological evidences, the comparative study of the embryology of different organisms shows that the striking similarity between them. To explain this phenomena, the biogenetic law was proposed by the Ernest Haeckel. And what this law says? This law says that an organism, it's in individual developmental follow, in its individual development follow the different developmental stages through which its ancestors have passed in the course of their evolution. What it means says is that if the man is the best organism, which is as per the ladder of species from the Aristotle. And if it suppose has the fishes, it has the snake, it has the amphibians, like for example, if all these are actually been the sequence in which it has followed, so when the baby is going to born, right, it is actually going to first be amphibians, then it is actually going to be the fish, and then it is actually going to be it will show the features like a bird, and then it will eventually be developed into the man. So what the Ernest Haeckel is saying is that the organism, its individual development follow the different developmental stages through which its ancestors have passed. So man has passed through multiple stages by which it has actually reached to this final stage. But when the baby is going to born, and that is what is exactly happened, right? When the fetus are born, they are actually swimming into the into the womb, right, into the that water, and then that they will behave like a fish, right, and eventually they will develop like amphibians, then fishes, and then eventually they will develop into the man, right. So this is what example is what it's showing here. It will showing the example of fishes, salamanders, tortoise, chicken, rabbits, and the man. So what you see here is initially it is actually going to have the fetus, which is actually going to form, it look like as the amphibians, and then it will actually going to develop into the fish-like, and then eventually it is actually going to form the baby in the case of man, right? So in another word, this law, what it also says is that ontogeny repeats the phylogeny, which means if you want to go with the development, it is actually going to follow the phylogenetic tree, right? The phylogeneity means what are the different organisms through which you have been evolved. So let's take an example of the development of frog, for example. In its developmental stage, it forms the fish like tadpole larva with tail, then fin and gill for breathing in water. It indicates that the frog is evolved from the fish-like ancestors. So that is a classical example where the tadpole or the frog is actually forming the species, like, so that the frog is for initially giving rise to the eggs, then those eggs are being fertilized, and these fertilized eggs are then if they are swimming into the water like a fish, and then eventually they will develop all the other kinds of appendages and other things, and then they will develop into the frog. Then we have the paleontological evidences. So what is the paleontology? Paleontology is the study of past life based on the fossil record. The paleontology studied the number and the nature fossil in the early rock, distribution of the fossil in the successive strata. Now the question is, what are the fossil and how it is formed, and provide the information about the evolution. The fossils are the remain or the impression of the ancestral organism preserved by the natural mean in some medium. For example, this is a fossil of insects into the amber, right? So the medium found with fossils are like sedimentary rocks, it could be amber, it could be asphalt, it could be volcanic ash, ice, peat bogs, sand, and the muds. What is the mechanism of the fossil formation? So during the formation of the sedimentary rock, the dead animal of the sea or the large lakes and the land carried out to the sea or large by the river sink down and get buried into the rock. So when there will be a rock is forming, so for example this is a rock, right, and it is accumulating the soil, right, it could possible that one dead animal is going to fall, and then it is actually going to again covered with that particular soil particle, and because of that it is actually, and since there will be no entry of oxygen, this particular organism is going to be preserved. Supply of oxygen is limited in this condition and prevent the decay or reduce the decay rate to minimal. So because it will not allow the decaying at a faster rate compared to what we could see in the open, it is actually going to give you an impression. So when you take out, it is actually going to give you an impression of that particular animal, or and if you take out that soil, it will actually going to give you the molds, right? So as a result, the animal remains preserved in the rock and have formed the fossils. The hard remains of the dead got preserved layer by layer in the sedimentary rocks. The fossil present in the deeper layers are earlier and the upper layer had more recent rocks. There are several different kinds of fossils. These fossils are distributed in amber, asphalt, ice, volcanic ash, pet bogs, storm dust and the sand dunes. So this is the table where I have shown all different types of fossils, their formation and examples. For example, there are fossils where the entire organism is being preserved. The example is the woolly mammoth in the Siberia, insects exoskeleton, mummies of the mammals and birds found in the California, and the giant alloc of Ireland. So these are the different skeleton. Like the other example is the skeleton material. So in this case, you cannot develop, you cannot have the entire organisms, but you can have the some part like the bones, teeth, shells, and all those kind of things. Then you have the molds and the cast. So here you will not get even the skeleton or the entire organism, but you will going to have the negative impression. So in this, the hard part trapped in the sediment and that hardened to rock, skeleton dissolve, leaving its impression as the mold, right? So this is a gastropods from the Poland. So that is an example of the fossil, and this is what is other examples of the fossils what is being present. So the question is how you can determine the age of the fossil. The age of the fossil can be determined by the following method. So in the past people were using the relative dating methods. So in the early dates the mechanism of absolute dating was not present, and as a result the relative dating technique were used to determine the age of rock and the fossil. In this technique the position and the erosion rate of rock in particular environment. The older rocks are situated in deeper, had ancient fossils, and superficial rock had the fossil of the recent fossil. So this relative dating technique is very, it's like you say that, if you have a rock, right, and if you find the fossil here, if you find the fossil here, since this is actually in a deeper portion and this is a shallow portion, this is going to be earlier fossil, this is going to be the late fossil. But this is all based on the relativity. This is going to be a relative. So that's why the people were looking for the absolute dating method, and they have developed the multiple absolute dating method. These methods are using the spontaneous decay of unstable radioactive nuclei into the stable radioactive nuclei at a constant and known rate. Absolute dating technique uses the radioactive nuclei in three different techniques. So you have the uranium lead technique. So this is the uranium decay what has been shown. So if you see the very clearly, in the uranium lead technique, this technique was introduced by the Baltwood in 1907. So the rock contains the uranium, which is the uranium-238 in the form of mineral zircon. So uranium decays spontaneously to lead as per the given scheme. So you have the uranium-238, then it will going to form the thorium-234, then it is going to form the protonium and so on, and ultimately what you are going to form is you are going to form the lead. So depending on the amount of lead, depending upon the amount of uranium, you can be able to identify how many life cycle, how many half-lives are being crossed, right? For example, if one half-life, the uranium is going to develop into thorium, but the difference is the 4.5 into 10 to the power 4 years. So if you count the number of half-lives, you can be able to count the age of that particular fossil. So it has a half-life of 4.5 billion year, which means it will take this many years to decay 50% uranium. So determination of the content of uranium and the lead in a rock or fossil can be used to determine the age of rock. For example, if I got 40%, right, or if I got the 50% decay, so suppose I got the total amount, and suppose I got 10 mg of uranium and 10 mg of lead, right? That means that there is a 50% decay, and that means that the age of that particular fossil or the age of that particular rock is 4.5 billion years, right? You can easily calculate accordingly the same way, right? If you got, like for example, if you got 2 mg of uranium and you got 8 mg of lead, then also you can be able to calculate, right? You can be able to calculate. So there will be 80% decay, right? So that's how you can be able to calculate the age of that particular rock using these kind of equations. Then we have the carbon dating method. So carbon method is also been introduced by the W.F. Libby in 1950. The radioactive carbon C14 is found naturally in rock, and C14 has a half-life of 5600 years, and C14 decay gives rise to the nitrogen 14. Carbon dating method can be used to measure the fossil age up to the 25 years age. So you can see that you have the uranium method, you have the carbon method, and you have the potassium method, and all these methods are actually having the multiple range in which you can be able to use. Then we have the potassium argon method. So radioactive potassium is usually found in the rock of alnite. It has a half-life of 13.9 years, and it is disintegrated to form the organ. So you can actually be able to use all these three methods to calculate absolutely. There will be no relative dating method. So these are absolute methods. It will actually going to tell you what is the age. Age is 10, 20, whatever, right? And that's how you can be able to correlate the different fossils what is being found even in the different areas or different region within the world. Now, utilizing what is the utility of determining the age of the fossil? That if you determine the age of the fossil, you can be able to utilize that information to calculate when that particular organism is being appeared. And based on this, the people have come up with the geographical scale or geographical time scale, right? Geological time scale is actually going to tell you the appearances of the different animals, and so use of the radioactive dating method has allowed the determination of fossils found in the different sedimentary rock sample. It has allowed to calculate the presence of different organisms preserved in the rock sample in the form of fossil. In addition, it helps the scientists to predict that the earth is approximately 4.5 billion years old and life appeared on earth almost 400 million years ago. So these two informations actually came because of the carbon dating or the absolute dating techniques. Since then, the earth's history has been divided into 5 different time frames known as the eras. Few of these eras are divided into periods and which turn in splits into the epochs. The different eras are as follows. So you have the Archeozoic era. So that Archeozoic era is starting from the 4600 to 3500 million years. Remember that it is always been done in the reverse order, right? You see the higher number and then you see the lower number, right? So this means we are talking about from the reverse, right? So it is the first era and began with the formation of Earth and presence of the solar system. There is no fossil form available during this era. This means this is the era when the Earth is actually being formed. Then we have the Proterozoic era, which is starting from the 3000 to 1000 million years. It is the second era and began with the origin of the prokaryotes, the primitive metazoans, and the eukaryotes. Reports are available about the scanty fossils in this era. Then we have the Paleozoic era, which starts from the 570 to 280 million years. You see, there is a gap actually, and these gaps actually, since there are no fossil found between this and this age, we cannot have any idea what is happening. So it is the third era and known as the era of ancient life. It saw the appearance of invertebrates, fishes, amphibians, and reports of spore-bearing animal plants, like which means trees, ferns, and origin of conifers is available. Initial reports are also available about the scanty fossils in this particular era. Then we have the Mesozoic era, and the Mesozoic era is from the 225 to 135 billion era. Disappearance of the tooth birds, right? Therian animals, reptiles, and the dominance of dinosaurs. In addition, the placental mammals are also found. The reports of cycads and the flowering plant is also available. And then you have the Cenozoic era or the modern era, which is starting from the 135 million years. This is the modern era and it witnessed the dominance of present age man, modern mammals, birds, fishes, and insects. So this is what we have discussed so far. What we have discussed about the evolutions. We have discussed about what are the different evidences what we have produced which actually support that there is a evolutions. So we have discussed about the morphological and structural evidences. We discussed about how the body organization is giving you the idea about the how the different organisms are evolving from the pre-existing environment. Initially you have the cellular level organizations, then you are having the tissue level organizations, and then you are having the organ or the organ system organizations. And you know, and we have discussed also the many advantage as well as the disadvantage of these systems. Then we have also talked about the homologous as well as the analogous organs, and we have said that the homologous organs is actually going to give you the idea about the diverge evolutions, whereas the analogous organs is going to give you the evolution about the converge evolutions. So if you could study the homologous as well as the analogous organs, you can be able to make the very clear and crystal pictures who appeared when, right? And then we also talk about the gradual modification, how the different organisms have adopted to the change environment, and because of that they have changed the physiology, they have changed the organs. We have taken an example of heart, and we have discussed about how the heart is being converted from the 2 chamber to the 4 chamber. And then we also discuss about the connecting links, and connecting link is a very crucial features or crucial evidence that proves that these two organisms are coming from that particular ancestor connecting links. And then we also discuss about the embryological evidences as well as the paleontological evidences. And at the end, we have also discussed about the different eras. We have also discussed about the geological time scales, and we have discussed about the different types of eras what are present. So with this, I would like to conclude my lecture here. In the subsequent lecture, we are going to discuss about the how the evolution is happening. So till then, goodbye. Thank you.